Hey there. The purpose of this video is to discuss protein structures and the factors that go into play with how a protein ends up getting the structure that it ultimately has. So there are 4 ways to categorize protein structures. And I'm just going to move my picture over a little bit. Maybe I'll put it down there. So remember that amino acids can come together to form, when they come together, they form a peptide bond, and you can have a dipeptide or a tripeptide or a polypeptide. To have it categorized as a protein, we need at least 50 amino acid units. So proteins, once again, serve very specific biological functions, and we can describe them on 4 levels. The first level is the primary structure, which talks just about the amino acids being held together in peptide bonds. Secondary structure includes the initial folding that starts to take place as the protein is being made, and the dominant force that holds secondary structure together is hydrogen bonding between hydrogen and oxygen on the protein backbone. And examples of those include alpha helices and beta pleated sheets. Tertiary structure is the overall conformation of the protein, and what determines it primarily is the nature of the side chains. And then quaternary structure occurs when we have more than one protein chain coming together and being held together with forces to produce a larger unit. So we have protein chains as subunits within a larger complex. So that was a couple-minute introduction with lots of words, and what's better than that is pictures, quite honestly. So I've got lots of pictures in this next set of sequences. So primary protein structure is just the amino acids being held together. Again, it's the actual sequence of amino acids, and a protein has a very specific sequence of amino acids. So hemoglobin, one of the subunits in the hemoglobin complex, always has the exact same sequence. And there are 4 subunits within hemoglobin. Secondary protein structures include that initial folding that takes place, and 2 examples are the alpha helices, which is like a spiral. Got to go in the direction that that's spiraling, and then a beta pleated sheet. Tertiary occurs when side chains are interacting with each other, and here within there you can see that secondary structure represented within the chain of amino acids, and then when we bring 2 together, it is considered 2 or more. It's considered quaternary structure. So amino acid sequence, again, is the primary structure. And here is an example of the get me out of the way. This would be 2 units and 2 subunits within insulin, and here the first is a glycine followed by ILE, whatever that is. I cannot remember. I think it might be isoleucine. We don't really need to remember this. And it affects all other levels of protein structural organization, because it is what is on these side chains that are ultimately going to determine those secondary and tertiary structures, primarily tertiary. Secondary structure is primarily held together by hydrogen bonding between the oxygen of the carbonyl group within the amino acid residues, and also the hydrogen of the amino group within those polypeptide chains. So an alpha helix an alpha helix looks like a spiral. And so we're not looking at side chains here as the primary function here. We're looking at this carbonyl group within this backbone here, hydrogen bonding with the positive portion of this hydrogen coming from the hydrogen, or the unequal sharing of electrons between nitrogen and hydrogen. So all of these little dots represent those hydrogen bonding interactions. And here is a beta pleated sheet. Again, notice that the R groups are not playing a role in this. It is interactions involving hydrogen bonding between the major backbone of that polypeptide chain. The tertiary structure is kind of the overall three-dimensional shape that the protein ends up taking. And there are lots of interactions that hold these tertiary structures together. We can get some hydrogen bonding between side chains. If we have the cysteines, we can get actually this disulfide bridge is a covalent interaction holding these two portions of this structure together. Here we have a salt bridge. So we have this NH3 group is on the R chain. It's a basic R chain. And this is an acidic R chain or side group. And when we get the forces between these positive and negative charges, it's referred to as a salt bridge. Oftentimes our non-polar groups, our hydrophobic groups, are going to be tucked towards the inside. And I'm not sure what that is. I may have to come back and watch that later because, or you may have to watch that later. I'm positive that I didn't set up so that you could hear it. Maybe there's no noise. Let's just click it and see what happens. I'll change my sharing settings. That shares the computer sound. There we go.
Structure is tertiary. Superimposed on primary and secondary structure is tertiary structure. Irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
And here's another example showing that tertiary structure. We've got some hydrogen bonding. Again, if we have an acidic side chain interacting with a basic side chain, those are ionic sorts of interactions. It's called the salt bridge, or hydrophobic, tucked towards the inside. Here we have some polar bonds. This is a pleated structure with hydrogen bonding occurring between those. So quaternary structure occurs between some more complex proteins. And let's look and see what this animation does for us. It might not do anything. We'll see. Oh, it is not going to let me do anything. So I will bring this back. Thank you for your patience. Here are some more examples, some colorful renditions. Primary structure, again, that chain of amino acids. Secondary includes alpha helices and beta pleated sheets. Tertiary is the overall conformation, and quaternary is more than one coming together. Again, some more pictures showing the same concept. I think the more pictures you can see showing similar concepts and ideas, the better it gets ingrained within your brain. And this is a true or false. All proteins have primary structure. In your mind, please commit to an answer. And the answer is true. It refers to the sequence of amino acids. Now, when proteins exist in their structure, sometimes that protein structure can become lost. That entire structure can become unraveled. So here's an example of that. Here is the overall structure of the protein, and this is an unraveling of that. So denaturation is the alteration or the uncoiling of secondary, tertiary, and quaternary structures. However, it does not alter that sequence of amino acids. So if this is the train car, each of those amino acids in this polypeptide, each of these residues are still held together. So when we denature a protein and it uncoils, it no longer has the same functions. This is another image representing that that uncoiling. Heat is something that can denature protein, and this is something that's desirable for us when we're cooking foods. Other things that can cause denaturation include mechanical agitation. I'll show you an example of that on the next slide. Detergents, organic compounds, pH changes, inorganic salts, and UV light can all unravel or uncoil those quaternary, tertiary, and secondary structures. However, they do not impact the amino acid connections, those peptide bonds. So here's an example of how we use this to our advantage. The proteins within an egg, when we cook them, become denatured, and that's why the structure of the cooked egg looks much different than that. When we take egg whites and we whip them into a froth like this, that is an example of a mechanical action that is denaturing those proteins to give us a very desired product. So the loss of primary structure, the separation of amino acids, is called hydrolysis. Remember, hydrolysis is the cutting, hydrolysis. It is the cutting or the cleaving of a molecule with the addition of water. So this destroys our primary structure. This occurs when we eat our proteins, and our bodies will, under the right conditions, break up those peptide bonds. So we have lots of individual amino acids floating through our bloodstream, floating through within our cells, floating to different parts of our body. So this produces free amino acids. It can occur in the presence of enzymes. It can occur in an acidic environment, and it can occur in a basic environment. So this bond gets broken, and what happens is the H and the OH get added back to those residues. Remember, the amino acids within the peptide bond are referred to as residues because it's what's left. They're missing the original hydrogen and OH group that were part of the amino acid, and now they're getting them back. So hydrolysis is the breakdown of proteins into individual amino acids. And just a little representation of what happens in our bodies. So we eat the protein, the amino acids are held together, these residues are held together with peptide bonds. They will break apart into the amino acids through hydrolysis, and then wherever these amino acids are needed, they will become parts of new proteins. So this is that, remember, when two amino acids come together, it's called a condensation reaction or a dehydration synthesis reaction. So this is just a very, very basic picture that shows what happens with amino acids in our bodies in the destruction of that primary structure and then the recreation of it in another protein.